This is a recording of Chapter 15, the digestive system. Over here is an overview of the different things that we eat. Those are your macromolecules. So we end up eating this big molecule of carb, whether it's starch or glycogen. That is a polysaccharide. It's too big to be absorbed as such, so we have to start breaking it down. We can also eat these two disaccharides, which is sucrose, and that is table sugar, or lactose, and that is milk sugar. Your intermediate product of the breakdown of this polysaccharide will be maltose. Again, our body can only, or the small intestine can only absorb a monosaccharide. So it'll break down the maltose using an enzyme, maltase enzyme, into two glucose molecules. Sucrase will break down sucrose into a glucose and fructose molecule, and lactose will be digested by a lactase enzyme into a glucose and a galactose molecule. These end enzymes, the maltose, sucrase, and lactase, those are found inside of the cells of the intestine. Those are known as intestinal enzymes. And then we have the macromolecule of a protein made out of this amino acid. Again, too big of a protein, so we have to digest it into smaller peptides, and we'll do that with pepsin, trypsin, and chymotrypsin. We'll end up having this three peptide chain, or three amino acid peptide chain, and we have to remove the amino acid on the carboxyl end, that is by the function of the enzyme carboxypeptidase, and then we need to remove the amino acid on the side of the amine group of the protein, and that's by an enzyme, amine peptidase enzyme. And then we have breakdown into 20 different kinds of amino acids. Those are the ones that we can eventually absorb. And then fat or triglycerides, we have a glycerol molecule and three fatty acid chains all combined together to make a triglyceride, which again is too big, and we are going to digest it by a lipase enzyme into one monoglyceride and two free fatty acid chains. So the function of the digestive system really doesn't, you don't want to think of it as only terms of digestion and absorption, but it all starts at the very beginning with the ingestion of food and liquids, and then we start the digestive process, and then there's absorption, and then the liver takes up most of our, everything that we absorb has to pass through the liver first. We kind of think of the liver as the kitchen of the body, where we'll take up all of the stuff that we just absorbed, the amino acids, the glucose, and so on, and do all sorts of different metabolic transformation of all of these molecules. It also has the ability to detoxify foreign substances. It can also eliminate different small amounts of metabolic, elimination of small amounts of the metabolic end products that are secreted by the liver. And we also have different immune cells. So the intestine has the scattered lymphatic tissue that are known as MALTs, or mucosa-associated lymphoid tissue, and those help in the production of antibodies and help in fighting infectious organisms as they try to enter the body. This right here is is a longitudinal section in the intestine, and you can see the four layers. There's the mucosa, and that's the layer that is in contact with the lumen of the GI tract, and then you have underneath it, you've got your submucosa layer, and then your muscularis layer, followed by this white serosa layer, which is basically the visceral peritoneum. So we'll talk about the mucosal layer first, and that's where we have the columnar epithelium with different cells that are secrete different things. So you have some mucus cells that make mucus. You also have these exocrine cells that make digestive enzymes, and we have the enteroendocrine cells, and those are responsible for making different hormones. So an enteroendocrine, so endocrine cell is going to make a hormone, and because it's found in the intestine, we name them an enteroendocrine cell. Your submucosal layer, that's where you have your blood vessels, and you also have in there these what are known as your submucosal plexus of nerves. That submucosal plexus, you see here that they usually end, that they're going to end at these enteroendocrine cells, and they are, the mucus cells or the exocrine cells. So these are basically when you stimulate that when you stimulate your submucosal plexus, that's going to increase secretions. Your muscularis layer, that's made out of smooth muscle fibers in 2 different layers, and you also have that plexus of nerves, and this one known as the myenteric plexus. And when you stimulate the myenteric plexus, that is going to stimulate motility. So in order to remember those, the submucosal with an S, when you stimulate that plexus, it is going to increase secretions with an S, while myenteric plexus stimulation with an M leads to increase in your motility. So how is the GI process regulated? Well, the gastrointestinal system is made that to digest and absorb anything you eat. Okay, so it really is not going to depend on the needs of the body. It is going to depend on what we eat. So it's designed to absorb all of the nutrients that are ingested, regardless whether the body actually needs them or not. Okay, and it's really governed by the volume and the composition of what we eat, rather again than the nutritional status of the body. So the GI reflexes are initiated by local things, for example, the distension of the wall by what we eat. So that wall stretch can stimulate motility and can stimulate secretions. The chyme osmolarity, so the total amount of salt that we eat, concentration of salt, the acidity of the chyme, and the chyme concentration of certain digestive products. So is it mainly starch? Is it mainly monosaccharides that we digested or that we absorbed? Is it mainly fatty acids or amino acids? So again, depending on kind of what we actually ate. The neural regulation of the GI process, we already talked about the myenteric and the submucosal plexuses. Those are known as the enteric nervous system. So we kind of talked about how stimulation of the myenteric will increase motility, stimulate the smooth muscles of the intestine, while the submucosal stimulation is going to lead to increased secretions. But there is also a higher level of control. So the autonomic nervous system, the sympathetic and parasympathetic, also have a control and say on what, to these myenteric and submucosal plexuses. So we have 2 different reflexes going on. There is a short reflex, okay, that is within the myenteric and submucosal plexuses, and we also have the long reflexes, and those come all the way from the sympathetic and parasympathetic neurons. So you can see here how these kind of reflexes work. Okay, you have certain things that will stimulate the autonomic nervous system, for example, hunger, okay, or smelling or seeing food or taste, just thinking about food. That is going to stimulate the parasympathetic part of the nervous system. And that parasympathetic is going to send impulses to the local nerve plexuses, so your myenteric and your submucosal plexuses, to stimulate motility, to stimulate the smooth muscles, and to stimulate the glands. And that would be your response. Okay, that would be your GI response to those, to that hunger or to that stimulation that happened. You also have, now that you eat, that whatever's in the lumen, that can also lead to stimulation of local receptors. So, the stretch, for example, can stimulate the mechanoreceptors that are found in the stomach or in the intestine. And that's going to send impulses to your nervous system that in return can also lead to more stimulation and more secretions to come out. For the hormonal regulation, we have the local hormones that come out of the small intestine. Those are known from your enteroendocrine cells. Those cells are pretty much scattered throughout the stomach and the small intestine. Okay, they are found on the side of the small intestine that is towards the lumen, or so towards the contents of what we're eating. And there are the four of these GI hormones that we're going to be talking about. There's gastrin, CCK, secretin, and GIP, or glucose-dependent insulinotropic peptide. So first off, we'll talk about gastrin. It's a peptide hormone. Actually, all of these are peptide hormones. The gastrin is made by the antrum of the stomach, and that's the lower part of the stomach. It is stimulated by the parasympathetic nervous system. It's also stimulated by proteins in particular. So when we eat proteins, amino acids, and having peptides in their stomach, that'll stimulate gastrin. Gastrin is going to stimulate and increase the acidity and the motility in the stomach. So gastrin that comes out of the stomach, okay, out of your, anything related to the stomach is kind of termed gastric. Okay, it will also increase your secretions of the stomach and gastric motility. Okay, it stimulates the small intestine motility and the movement of the large intestine. That gastrin is responsible for a reflex known as the gastrocolic reflex. So from the name, the reflex starts in the stomach and ends in the colon, where some patients will come and tell you that we are unable to have a bowel movement unless we have something to eat. And that is because of the effect of gastrin. So when food gets into the stomach, gastrin is released by the stomach. It will increase the motility of the large intestine, leading to that bowel movement. Okay. The factor that can inhibit the release of gastrin is somatostatin. Now, CCK, which is also a peptide, comes out of the small intestine. So CCK, secretin, and GIP, these are all released from the small intestine. The only one out of the four that comes out of the stomach is gastrin. Now, CCK, secretin, and GIP, that is going to be released when the stomach is starting to empty its contents. So as that chyme is now passing through the duodenum, that will lead to the release of these hormones. So CCK is released when the chyme is made out of mainly amino acids and fat. Okay, so you'll see here that it'll inhibit the acid secretion and motility from the stomach because we are done with gastric secretions. We no longer need it. The stomach is already emptying its contents into the duodenum. So these hormones are going to come out and tell the stomach you can stop making HCl, you can stop your motion or your motility, you're already emptying your contents. CCK is going to stimulate insulin secretion from the pancreas. It also stimulates the contraction of the gallbladder and the relaxation of the sphincter of Oddi, which is the sphincter, it's the hepatopancreatic sphincter that releases bile and the pancreatic juice into the duodenum. Secretin is a second peptide hormone that comes out of the small intestine, but this is stimulated by the acid that is coming out of the stomach. So by negative feedback, it's going to inhibit acid secretion, and it's also going to inhibit gastric motility. Okay. And it will stimulate bicarb secretion by the pancreas. Okay. Your GIP hormone is the last of the peptide hormones that come out of the small intestine, and that is going to, the stimulus for release of GIP is glucose and fat in the small intestine, and it will stimulate the release of insulin from the pancreas. Here is how CCK works. So we'll start off with the chyme that's being released from the stomach into the duodenum. Okay, that is going to stimulate the enteroendocrine cells of the duodenum to make CCK. CCK will go through the bloodstream and reach the gallbladder. When it reaches the gallbladder, it'll contract the gallbladder, lead to the contraction of the gallbladder and the relaxation of the sphincter of Oddi. In order to control the phases of gastrointestinal control, we have three different phases. There's cephalic, gastric, and intestinal phase. So cephalic phase, that is mainly under the control of the autonomic nervous system. So again, seeing food, hearing, or thinking about food, all of that is going to stimulate the parasympathetic nervous system. Those impulses are carried by the vagus nerve, and that will stimulate both the motility and secretions by the GI tract, kind of preparing your digestive system to receive food. The gastric phase, okay, that's now where food is now within the stomach, so there's that distension of the stomach, stimulating the stretch receptors. It also depends on what we ate. So was it mainly protein? Was it fatty acids or fat? Was it carbs? Okay, so we have these, these are all mediated by the short and long neural reflexes and by the release of gastrin. The intestinal phase, though, that's when, food is now going into the small intestine. That phase is also mediated by both your short and long neural reflexes and by the hormones, the rest of the hormones that we talked about, secretin, CCK, and GIP. Here you can take a look at the intestinal phase. So this is where we're going to start in this flow chart. So in the duodenum, we're going to get acidity from the hydrochloric acid that is coming from the stomach. There's also fat, amino acids, there's salt, and there's the distension of the wall of the small intestine. All of that is going to stimulate your enterogastrones, to stimulate the duodenum to make CCK, gastrin, and GIP. It is also going to stimulate the short reflex and longer reflexes through the parasympathetic. That would lead, actually, now that food has reached the small intestine, which is pretty much, one of the later stages of digestion, you'll start to inhibit your parasympathetic and stimulate the sympathetic. Okay, so in this course, we really don't get into the breakdown of the basic structure and function of the different parts of the GI tract. I just have it here to kind of, so that you guys could take a look at it. You can go over this maybe from previous courses or read it from your text. So again, I'll leave this up to you guys. This is a more advanced course on that than that. But we will talk about the physiology and the different functions of different parts. So, for example, saliva, these are the different components of saliva, and we want to talk about the function, what those components are and the function of each. So water, that is there to moisten food, and in order for the taste buds to be stimulated, in order to stimulate those chemoreceptors, those food molecules have to be dissolved in water in order to stimulate your chemoreceptors. There's also mucus, and mucus is there to lubricate food, and it'll help stick all of that food together into a bolus of food so that we can swallow it. There's bicarb. Bicarb is there to neutralize any kind of acidity in the food, and it also protects the teeth from, overnight when we're not eating, for example. No matter how much you brush your teeth and floss, there's always going to be, food particles in there that can be fermented by bacteria. So in order for us to protect ourselves, there is bicarb that can kind of neutralize that and protect us from that. Lysozymes, these are different enzymes that kill bacteria as well. Then we have these two different enzymes. These are digestive enzymes. So there's amylase, and that begins the digestion of starch. And there's also lipase, and that begins the digestion of triglycerides. So you see here that we really start the digestion of starch and fat in the oral cavity. Protein does not start in the oral cavity. Protein is going to start in the stomach. So what is the mechanism of swallowing? That swallowing center is found in the medulla oblongata, and it is all initiated when that bolus of food is pushed by the tongue all the way towards the back of the oral cavity, so into the oropharynx. That is when that reflex is stimulated. If you look at this part here, There are several ways the food can pass. So the food could pass down into the trachea, or it could pass down here into the esophagus, and that's where it should be going, or it could pass up here into the nasal cavity. So to make sure that food really only has one pathway to go, we have to close those other two pathways. And those closures are part of this reflex. So by reflex, that soft palate elevates and shuts down the back of the nasal cavity, while the epiglottis, and you can see that here, the epiglottis bends down, as you can see in this picture over here, bends down in order to shut and protect the trachea. So in this, now the food only has that one way to go, and that would be down the esophagus. Now here's a really fast breakdown of the anatomy of the stomach. Okay, so we have the esophagus, and then the stomach. There's a greater curvature and a lesser curvature. The upper part of the stomach is known as the fundus, and that's the dome. And then the body, which pretty much makes the majority of the stomach. And then there's the antrum, and that's the lower part, and followed by the duodenum, which is part of the small intestine. At the entrance between the esophagus and the stomach, there is a lower esophageal sphincter, and there is another sphincter at the exit known as the pyloric sphincter that controls emptying. The fundus doesn't really secrete much. Okay, it's there for expansion and kind of, for more space. The body secretes mucus, pepsinogen, and hydrochloric acid, while the antrum secretes also mucus, pepsinogen as well, just like the body, but in addition to that, it releases gastrin. So remember, the gastrin hormone that we talked about is made by the antrum of the stomach, as we've seen in that table. The antrum has very thick, smooth muscle fibers, and that is going to help in the mixing of the food with the gastric juice, in that grinding motion that happens. If you take a look at the microscopic makeup of gastric glands, we can see different kinds of cells. So I'll talk about these cells, and these cells are not really shown on this image, but we've got G cells that make gastrin, and there's D cells that make secretin. We have other cells, and those cells are shown here. We've got mucus cells, and that is obviously going to make mucus. As you go deeper, you have these green parietal cells. Obviously, they're not color-coded for real, but your parietal cells make the hydrochloric acid, and they make intrinsic factor. Intrinsic factor is needed to absorb vitamin B12. Okay? And in patients that have a disease known as pernicious anemia, that is an autoimmune disease, the patient makes antibodies against these parietal cells, destroying them, so their bodies will not be able to make intrinsic factor, and they will not be able to absorb vitamin B12. Vitamin B12 is important to make healthy red blood cells. So when they have vitamin B12 deficiency, they will not be able to make healthy RBCs, and they have a very resistant kind of anemia known as pernicious anemia. When you go a little bit deeper, we have these ECL cells. ECL stands for enterochromaffin-like cells, and those secrete histamine. Histamine acts as a paracrine hormone that is going to stimulate your G cells to make more gastrin. And gastrin, as we've learned, increases your acidity. So, in short, histamine can increase the production of hydrochloric acid in the stomach, and that is why it was found that some patients that take antihistamines to treat their allergies, as a nice side effect, it also helped them with their heartburn. In the very bottom of the gastric pit, or the gastric gland, we have these chief cells, and those make pepsinogen. Pepsinogen is an inactive hormone that has to be activated. And we'll see how that process happens over here. So this is your chief cell that's making pepsinogen. That's the parietal cell that makes hydrochloric acid and intrinsic factor. It's the hydrochloric acid that activates pepsinogen into pepsin. And pepsin is a protein digesting hormone So it'll see here that it'll start the process of protein digestion into smaller peptides. Any hormone that, on any enzyme that digests protein comes out in an inactive form. So you'll see that recurring throughout, throughout all of these protein digesting enzymes. Now, how does the parietal cell make hydrochloric acid? So remember this cell over here, that green cell that makes HCl. We're going to learn how that process works. So this again is your parietal cell that has two sides to it. This is the side of the stomach, towards the stomach, that's known as the apical membrane, and this is known as your basolateral membrane, and that is towards the deeper part of the body, so towards the capillaries. What this cell wants to do is to secrete HCl here into the gastric cavity. So what it does, it takes carbon dioxide and water molecule and it combines it to make this carbonic acid, or H2CO3. That is done by an enzyme known as carbonic anhydrase enzyme. This reaction is going to haunt us several times actually in this chapter, and then it'll come back in the renal system and respiratory system as well. So be very comfortable with this reaction where carbon dioxide binds with water through this carbonic anhydrase enzyme in order to make carbonic acid. Now, carbonic acid is an unstable acid. It will break down into two components, which are hydrogen and bicarbonate. Okay, so hydrogen, that's what we're looking for. Remember, we're making HCl. So hydrogen can be pumped out of the cell using what is known as a hydrogen potassium ATPase pump. So you see the process needs ATP. We'll break down ATP into ADP and use that energy to pump hydrogen out, and in return we have to bring potassium in. So that is known as your hydrogen potassium ATPase pump. So how do we get the chloride? Well, this bicarb is a negative ion. It'll be pumped or transported through this transporter. It'll be pumped out of the cell, and in exchange we'll take another negative ion, which is chloride. That chloride will go through the apical membrane, diffuse through these little transporters. The chloride, we've got chloride out here and hydrogen as well, and those are going to join forces and make hydrochloric acid. So the whole process of making hydrochloric acid really depends on the activity of this hydrogen potassium ATPase. So if you want to increase the production of HCl, you have to stimulate this enzyme. And that's what this is showing you. Again, it's all about the hydrogen potassium ATPase. And if you stimulate that enzyme, you're going to make more hydrochloric acid. And these hormones, these three hormones, gastrin, histamine, and acetylcholine, all three of these stimulate that enzyme. The only one that inhibits it is somatostatin. So how do we control hydrochloric secretion? Again, 3 phases: cephalic phase, meaning that we have not eaten yet, we are thinking or seeing food or smelling food. That is under your parasympathetic. So parasympathetic is going to stimulate the hydrochloric secretion through acetylcholine. Gastric contents, that is your gastric phase. So the distension of the stomach, especially if we're eating lots of protein, that is going to stimulate your long and short neural reflexes, and also by the release of gastrin. That is going to increase your hydrochloric secretion. So going back to that gastrin hormone, it'll stimulate this enzyme. Now the intestinal phase, and this is the phase where we're done with the stomach, food has gone into the intestine, has moved on. Now we no longer need hydrochloric acid. So you have your long and short neural reflexes and the enterogastrones like secrete and CCK. Those are going to inhibit your hydrochloric secretion. Now, how is it that we kind of tell the stomach to stop moving because we are going to start eating? Okay, that process is known as receptive relaxation. It is initiated by swallowing, okay, and the stimulation of the parasympathetic nervous system. That is going to release through these local neurons in the small intestine. It'll release nitric oxide and serotonin. Both of these are smooth muscle inhibitors. So that is going to inhibit the smooth muscle so that it can relax and receive the food. Once we're done eating, the sphincters shut down, the esophageal, your pyloric sphincter shuts down, and then there's a little pacemaker in the stomach that is going to initiate these strong contractions so that we could mix all of the gastric contents together. And it doesn't fire too much, so it's only, 3 contractions per minute. Okay, these peristaltic waves are going to happen. The antrum, as I said, which is the lower part of the stomach, has a thicker, smooth muscle, so those have very powerful contractions. At the end, when everything is all mixed up and we're ready to release the contents, the pyloric sphincter is going to relax, and we are going to start the emptying of the gastric contents. So we're going to move on into the small intestine. We want to kind of take a look at what the small intestine looks like. So we are going to move on to the picture actually. So here we have these circular folds. Everything that we're going to talk about here is in order to increase the surface area and increase the contact between the chyme passing through in the small intestine and between the chyme and the small intestine. Okay, so all of these are there to increase your surface area or the surface contact between them. And those are your circular folds that are found throughout the small intestine. Okay, so you can see here that circular fold. On top of the circular fold, you have these villi. Okay, each villus, if we take a look at this villus right here, each villus is made out of connective tissue in the middle with that lacteal. That green tube in the middle is a lacteal, and that is going to help with the absorption of fat. And you can also see, the artery and the vein. The villus is lined by simple columnar epithelium with microvilli. So you have your circular folds, you have your villi, you have your microvilli, and all of that is there in order to increase contact, to increase the surface area as much as possible. You also have these little intestinal glands. In the intestinal glands, that's where you'll find your enteroendocrine cells that secrete your hormones. We also have these what are known as Peyer's patches or lymphatic nodules. Those are the scattered lymphatic nodules that you have through the small intestine to try to trap anything that is trying to get absorbed with the food. And a little bit about, really quick about the structure of the ducts. Okay, so we have your liver also functions as an exocrine gland, okay, since we've got all of these ducts to exit. One of the things the liver produces is bile, and bile is going to be drained through the right and left bile duct into the right and left hepatic duct into the common hepatic duct. And then bile is going to be stored in the gallbladder. So know that bile is made by the liver cells, but it is stored and concentrated in the gallbladder. When needed, the gallbladder will contract by the action of CCK, and the bile is going to be released into the cystic duct and then into the common bile duct. The common bile duct is going to combine with this major pancreatic duct, and they both open at the same opening in the duodenum, which is your major duodenal ampulla or the major duodenal opening, and controlled by the contraction and relaxation of the sphincter of Oddi. So here we're going to be talking about the pancreatic secretions. Pancreatic doubles up as an exocrine, and an endocrine gland. So we're going to be talking about the exocrine portion. The exocrine portion makes your bicarb and some digestive enzymes. And we'll kind of take a look at the image right here. Okay, this part represents the acinar cells that are going to secrete your enzymes. And then you have this duct part, and that duct part is going to secrete bicarbonate. Okay, so going back to the parts of your pancreas, or the structure of the pancreas, we have the islets of Langerhans, and those are the endocrine cells. Those make your insulin and glucagon. That's not what we're going to be talking about. We are talking about the exocrine function, where it's made out of these acinar cells, and those cells secrete the enzymes. It is stimulated by CCK, while this duct portion of the gland secretes bicarbonate, and that is stimulated by secretin. Now, how do the pancreatic cells make bicarbonate? That again is by that infamous reaction that we talked about earlier. So carbon dioxide combining with with water, under the effect of carbonic anhydrase enzyme will make H2CO3, which is carbonic acid. H2CO3 will then break down into hydrogen and bicarb. In this case, now that we're actually making bicarbonate, this is what we want, but we have to get rid of the hydrogen. So we'll do that through a sodium-hydrogen exchanger. So we have to kick hydrogen, which is a positive ion, bring in another positive ion, in this case sodium, and then we'll kick out sodium using the sodium-potassium ATPase pump. And so we'll get rid of sodium, bring in potassium, but potassium has the ability to just diffuse out of the cell. It's the bicarb again that we are looking for. So the bicarbonate needs to get out into the duct of the pancreatic duct, and we will do that. We have to exchange the negative ion, the bicarb, with another negative ion, which is chloride. So we have what is known as a chloride bicarbonate exchanger. We will kick out the bicarb, take in chloride. Now this negatively ion chloride has to leave the cell, and it will do so through this channel known as the cystic fibrosis transmembrane conductance regulator, or CFTR. Now this is the same this is the gene, or the gene that controls the making of that transporter, or that regulator, is the gene of cystic fibrosis. So patients with cystic fibrosis have respiratory problems, and they also have problems with their pancreatic secretion, with the secretion of bicarb, and that is because it also affects this chloride transporter. Now, your pancreatic enzymes, lots of them digest protein. So as we said, they are going to be released in an inactive form. Okay, so you have for example, trypsinogen has to be activated into trypsin, and that is done by these enterokinase enzymes. So when the pancreas releases its protein digesting enzymes into the duodenum, they are in an inactive form. They have to be activated by the cells of the duodenum. These cells have enzymes known as your enteroenzymes, and these enterokinases are going to activate them. So one of those examples is trypsinogen being activated by that enterokinase into trypsin. Now this is a list of your pancreatic enzymes. So trypsin, chymotrypsin, and elastase enzymes, all of three of these are going to digest proteins. It'll digest the protein into smaller peptide chains. Carboxypeptidase is another protein digesting enzyme, and it removes the amino acid on the carboxyl end of the peptide chain. Lipase is an enzyme that digests triglycerides, and it'll remove the two free fatty acid chains and end up making two free fatty acids and one monoglyceride. Another enzyme is amylase, and that is going to help in the digestion of polysaccharides or carbs, splitting it into maltose. The last pancreatic enzymes are ribonuclease and deoxyribonuclease, and those break down nucleic acids, and it breaks it down into nucleotides. Now, so we talked a little bit about the secretion of bile, but we did not really talk about what bile is. So we said that bile is made by the hepatocytes, and it is stored and concentrated in the gallbladder, and then it has to be released into the duodenum. Bile contains bicarb, it contains cholesterol, phospholipids, bile pigments that gives bile its green color, and lots of waste products and bile salts. Okay, so we're going to be talking about bile salts. Again, the body is a very conservative thing. It does not like getting rid of bile salts. Okay, so it'll release these salts in the bile, but it will reabsorb it back from the intestine. That process, that recycling of bile salts, is known as the enterohepatic circulation. So we are going to see how that circulation works using this picture. So your liver makes the bile salts, and the bile salts, go through the right and left hepatic ducts. As I said, they are stored and concentrated in the gallbladder, and then when needed, by the action of CCK, now that we squeeze the gallbladder, the bile is going to be released through the cystic duct and then the common bile duct into the duodenum. Bile salts will perform their function, which we'll talk about in a minute. But again, the body will recycle them. So through the small intestine, we are going to reabsorb all of that bile salt, take it back to the liver via the hepatic portal vein. We lose about 5% of our total bile salts in feces, which is not bad. Okay, so the liver would have to make up for that 5%, but we tend to recycle 95% of the bile. Now we're going to break down the digestion of each nutrient. So first off, for carbs, the digestion of carbs starts in the oral cavity by an enzyme known as salivary amylase enzyme. Okay, but as soon as we swallow, it is deactivated by the hydrochloric acid that's found in the stomach. Okay, most of the carb is going to be digested in the small intestine by pancreatic amylase. The products of either salivary or pancreatic amylase would be maltose. Remember, we have two other disaccharides that we eat as disaccharides, and those are sucrose and lactose. So we have to break down now these three disaccharides that we have, namely maltose, sucrose, and lactose, into their monomers. We have to break them down into monosaccharides, and that happens by the enzymes that are found in the small intestine, known as their brush border. When we break them down into these monosaccharides, those can be transported, and those are the ones that we can absorb into the circulation. So here you'll see how this is the lumen of the small intestine. So we broke down the polysaccharides into maltose, and then we have your brush border enzymes, maltase enzyme that will break it down into monosaccharides. This right here is the intestinal cell, and this is the intestinal, that's the interstitial fluid. Okay. This is known as your apical or your brush border membrane, and we have here, that's your basolateral membrane. Again, this is the lumen of the small intestine, so this is where all of the chyme is passing through the small intestine, and this is where we need to absorb those sugars and transport it into the bloodstream. So all of these, oops, I'm in the wrong one, I'm sorry. So we're going to absorb all of these monosaccharides, fructose, glucose, and galactose. So fructose can go through this glucose transporter, okay, and that is on your apical or brush border membrane. While glucose and galactose, they come in together through a sodium glucose transporter. Right? Now that all three of them are inside of the cell, they can all diffuse through this glucose transporter into the interstitial fluid, eventually into your bloodstream. As for the digestion of proteins, that is going to start in the stomach by the enzyme pepsin, and then in the small intestine, the enzymes trypsin and chymotrypsin are going to, that are made by the pancreas, are going to digest proteins further down into smaller peptides until we end up with a peptide made of 3 amino acids, one amino acid in the middle, and then there's an amino acid on the carboxyl side and another on the amino side of the protein. And we have to break those down as well, so we'll do that with a carboxypeptidase and an aminopeptidase. Those are found in, again, the brush border of the small intestine. So how is it that we are going to absorb these amino acids? And we'll take a look at how that happens. From your apical border, which is the border that is facing the lumen of the intestine, these amino acids are going to be transported by secondary active transport coupled with sodium. And at the basolateral membrane, which is the side that is facing towards your interstitial fluid and the blood vessels, that is going to happen by facilitated diffusion. Infants have the ability to absorb whole proteins without even having to digest them by transcytosis, just like, for example, the absorption of antibodies that are found in breast milk. So this is, again, the intestinal cell. This right here is the intestinal lumen, where the amino acids are. And this is the interstitial fluid with the blood vessels where you want the, which will be your end destination after you absorb them. So you can see here your amino acids break them down by these different peptidase enzymes. We end up making these amino acids through a sodium transporter, sodium cotransporter. We transport the amino acids into the cell across that apical or brush border, and then we end up, there are amino acid transporters that allow the passage of these amino acids into the interstitial fluid. For the digestion of fat, so digestion of fat starts in the oral cavity by the action of salivary lipase, and then continues in the stomach using gastric lipase. But the majority of fat fat digestion really occurs in the intestine under the effect of the pancreatic lipase. And to split, all three of those enzymes assist in splitting your triglyceride into 2 free fatty acid chains and 1 monoglyceride. But in order for that to happen, molecules are usually big, and we have to emulsify them into smaller droplets. And that happens by the action of bile salts. This is where the bile salts that are made by the liver and the phospholipids come into action. Okay, so what we are going to do, when it comes to triglycerides, the digestive system is a little bit weird about that. What it does, it'll take these fat globules, break it down or emulsify it into smaller fat droplets or fat droplets, and then it will digest it into monoglyceride and 2 free fatty acid chains, absorb it as such into the intestinal cell, and then inside of the intestinal cell, it will reconstruct it back by the smooth ER, it'll reconstruct it back into a triglyceride, and then it's transported in our bloodstream as a triglyceride. So this is what, the whole process. you have, this is the fat globule that is in the intestine or that we ate, and we want to digest it. In order to do so, we have to emulsify it into smaller emulsion or smaller fat droplets. And that is, we emulsify them by the action of bile salt and phospholipid. So these bile salt and phospholipid are going to break down your fat globule into smaller emulsion droplets. We have not digested these fat droplets yet. Okay, so you can see here again, this is your droplet with the bile salts and the triglycerides that are, helped in the emulsification process. And then by the action of lipase, we were able to digest it into a monoglyceride and 2 free fatty acid chains. And then they are going to diffuse into the intestinal cell. Okay, if you take a look at this side, that is the same process. Okay, so in the lumen of the intestine, the fat droplet is going to, has to be emulsified. Okay, and we could see here after the emulsification process happened, by the action of lipase, we broke them down into your monoglycerides and 2 free fatty acid chains. They have diffused into the intestinal cell. Inside of the intestinal cell, we are going to reconstruct or remake the triglyceride in the smooth ER. Okay, so now you can see here that is the droplet of triglyceride again, reconstructed inside of the intestinal cell, and it is surrounded by some protein. Okay, in order for it to be able to be transported in the body. Proteins, or reconstructed triglyceride surrounded by protein, is known as a chylomicron, and it is absorbed not into the bloodstream, but it is absorbed into a lymphatic capillary known as the lacteal. Forgot to mention here that with the absorption of fat, we also absorb fat-soluble vitamins, which are vitamin A, D, E, and K. So now we're going to move on to our vitamins. So we have fat-soluble ones and water-soluble vitamins. Fat-soluble vitamins, as I mentioned, are A, D, E, and K, and those are going to be absorbed along with fat absorption, just with the other lipids. And as a side note, celiac disease is an autoimmune disease where the body makes antibodies against the intestinal brush border, and that process is induced by ingestion of gluten. So if, when these individuals eat gluten, that stimulates their immune system to make antibodies against their own intestinal brush borders. Okay. Water-soluble vitamins, those are absorbed by diffusion, except for vitamin B12. So remember we kind of mentioned vitamin B12 where we said we need intrinsic factor, and intrinsic factor is made by the gastric mucosa. Okay? In the absence of intrinsic factor, the body is unable to absorb vitamin B12, and we named that condition pernicious anemia, which is also another example of autoimmune disease where the body now makes antibodies against its own parietal cells, or maybe that patient got a gastrectomy for maybe, for a possible, could be cancer, for example, that they had gastric cancer or stomach cancer. They had to remove the stomach, so now they've lost all of their parietal cells and they've lost their intrinsic factor. For the absorption of water and minerals, minerals are absorbed by diffusion, and water is just going to follow by osmosis. Okay, most of the absorption of water and minerals happens in the small intestine. Okay, so the absorption of sodium, chloride, and potassium, calcium, and iron, for example, those there is a lot to do with the homeostasis of these different ions, and depending on the needs of the body, we can either increase or decrease the absorption of these ions from the intestine. So we're going to take iron as an example. So what happens to iron after we absorb it? So iron will get into your intestinal cell or the enterocyte, and it can be stored there. It'll bind to a protein known as ferritin, okay, and it'll be stored inside of the intestinal cell in case we need it in the future. So let's say somebody is bleeding and they need iron to make new RBCs. They can get that iron from the intestinal cells, again, from that storage. Iron that does not bind, okay, or that is absorbed into the bloodstream can circulate attached to another protein, and that would be known as transferrin. Okay, so ferritin is how we store iron. It's not really just limited to storage in the enterocytes. We can also store iron in our hepatocytes, so in the liver as well. Or iron can be freely floating in our bloodstream, but it has to be transported with a protein as well, and that protein is known as transferrin. There's a genetic condition known as hemochromatosis, and that would be there's a defect in the homeostasis of iron where there is accumulation of iron in your body tissue. For example, as we said, instead of being stored in limited amounts in the enterocytes or in the hepatocytes, they are now massively stored there, and that leads to damage of these organs. The treatment of this condition is frequent phlebotomies, or frequent blood donations. Okay, so where these patients will get rid of, with each blood donation, they're going to get rid of lots of RBCs, and so all of that stored iron is going to be released in order to make new RBCs and to replace those. Now we'll talk about the hepatic portal system, or the hepatoportal circulation. Everything that is absorbed from the small intestine, all of the venous blood coming from the small intestine, from the pancreas, from the stomach, and from the spleen as well. It's not shown in here, but from the spleen too. All of that has to go into the liver. All of that venous blood, before it reaches your systemic circulation, before it goes to the heart, it has to go through the liver first. Okay, so we are going to deliver all of the nutrients that we absorb from the intestine into the liver. We'll deliver the insulin and glucagon that were released by the pancreas. And the liver is going to use all of these nutrients to make different things. Okay, so again, you want to think of the liver kind of as the kitchen that is going to get all of the ingredients, the amino acids, the carbs, the monosaccharides, and take those and make different things in the liver. Okay, it can also detoxify anything harmful that we accidentally ate too. Now, how do we start eliminating different things? When we're done eating, okay, and we're done digesting, and after things, so when we're done digesting all of the products, we have to make sure that the small intestine is empty. Okay, if there are any remnants of undigested substances that remain in the small intestine, they can ferment, and that can lead to the overgrowth of bacteria. So we have what are known as migrating myoelectrical complexes, or MMCs, that make sure that we literally squeeze every single thing out of the small intestine and push that into the large intestine. These strong, powerful contractions, strong and powerful relatively to smooth muscles, okay? So it's nothing that we usually feel. Those are, but these are very different than the peristaltic waves that occur during digestion. Okay, these again, you kind of want to think of it maybe as having an empty tube of toothpaste, or you're trying to press and make sure that you're running out of it, you're trying to get anything out as possible. That is what the small intestine does. It literally squeezes itself to get everything out. It doesn't run throughout the whole small intestine in one setting. It goes kind of 2 feet at a time. Okay, and the whole process from the duodenum all the way down to the end of the small intestine can take about 2 hours. Okay, so again, this is there in order to make sure that anything that has not been digested or absorbed, that it moves on to the large intestine in order to go become defecated and prevent the overgrowth of bacteria. So what happens in the large intestine now that everything has been pushed by these MMCs into the large intestine? There are no digestive enzymes in the intestine. So if anything reaches the large intestine and has not been digested, well, it's out of luck because we do not have any enzymes there. Okay, the only secretions that come out of the large intestine are mucus, and that is kind of there to lubricate because most of the water has already been absorbed. And water absorption happens even more in the large intestine. So you want to kind of lubricate to help in the defecation process by the addition of mucus. And there is some fluid that comes out that has bicarb and potassium. Okay, the colon helps in the homeostasis of bicarb and potassium by having that ability to secrete them. The only thing really that the large intestine can absorb would be a little bit of salt followed by water, by osmosis. Some of the fibers, I'm sure you guys have heard that eating fiber is healthy. The thing about fibers is that these polysaccharides, that we do not digest them, okay, but they are going to be converted to short-chain fatty acids and gases by the bacteria. These short-chain fatty acids are important. There's lots of recent research that show that these fatty acids are important for the function of the healthy function of the immune system, cardiovascular health, and the function of the nervous system. There is a video that I posted for you guys about this on Moodle, and it is, so I need you guys to watch that video and post a little something on the discussion, and that is for extra credit. Now the bacteria that some of these bacteria also produce vitamin K, so we have that ability to make our own vitamin K that can be absorbed into the body. So what about the motility of the large intestine, and what about that mass movement or bowel movement? This is very different from one person to another. Sometimes some individuals have, 3 bowel movements a day, others have it once a day, it all depends. They're very rapid movements that happen, over the ascending, transverse, and then down to the descending colon. Usually when the rectum starts to dilate that starts to distend, stretch because of the contents reaching the rectum, this is when the defecation reflex is initiated. The defecation reflex will lead to the relaxation of the internal anal sphincter and the contraction of the sigmoid colon. I want to see if there's a picture of those parts. Yeah. So contraction of the sigmoid colon, the rectum, and the anal canal. Okay, and that will push all of the contents out of the body. And that concludes chapter 15.